everyone to the third of the RD-Connect webinars for the European Reference Networks. So today we are going to focus on fair data, what it means, why it's important that data are shareable and interoperable in databases in rare disease research. So just some organization stuff. So the presentation is going to take roughly 30 minutes, right, Marco?
Yes.
Yeah, something around 30 minutes, and then we'll have time for questions. So if you have any questions, please type them in the chat box. So you should see a bar on the bottom of the screen, and one of them is of the buttons is labeled chat. So please type in your questions in there so that everyone stays muted during the presentation, so we can do it without interruptions. And before Marco starts, I'm going to make a very brief introduction of RD-Connect for those who maybe would like to learn more about generally about what we do. So give me a second, I'll share the screen. Oh, that's not what I was going to share. That's interesting. Once again. Can you see my screen?
I can.
Okay, that's great. So today we're going to talk about fair data, but first I'm going to explain a bit more about RD-Connect. So we are dealing with rare diseases. What's difficult about rare diseases is that the patients are so few and they are scattered across countries, across the world. So it's very difficult to do any research or even provide any healthcare. That's why it's very important that in case of rare diseases that we do international collaboration and that we share and exchange and pool data, which will allow us to do better research, diagnosis, run clinical trials, and provide help to the patients. So currently there is a lot of research going on rare diseases, but a lot of data is generated, but those different types of data are stored in different databases, and usually they are siloed without much connection between those different databases. And that's why RD-Connect aims at connecting those different types of data and making them available to researchers and clinicians to improve research and healthcare. And for this reason, the European Commission has has funded RD-Connect to enhance and promote and enable data sharing in the rare disease research. And when I speak about data sharing, I mean sharing of genomic data, phenotypic data, as well as information about patient registries and biosamples in biobanks. So for this purpose, RD-Connect has created an integrated platform which consists of 3 systems. The genome-phenome analysis platform, which lets researchers and clinicians analyze genomic data, diagnose patients, and discover new rare disease genes. A sample catalog, in which you can find information about individual biosamples stored in rare disease biobanks. And the registry and biobank finder, which contains information about different rare disease registries, patient registries, and the type of information they have in there. And how does this work? RD-Connect receives data from researchers and clinicians, for example, those in the European reference networks, as well as biobanks, about the omics data, phenotypic data, biosamples data, and all that data goes into the integrated RD-Connect platform, where it's made available for researchers and clinicians to improve gene discovery, diagnosis, and therapy development. So, of course, this is very beneficial for the clinicians and researchers, but most importantly, it's beneficial for the rare disease patients. RD-Connect has the RD-Connect project is currently coming to an end next month, but RD-Connect as such is going to continue working in a different form. So very recently we have launched the RD-Connect community, which is an association of individuals or organizations or research groups who have the common aim of supporting and promoting data sharing in rare disease research. So this community, the membership is open free of charge for anyone who is involved in rare disease research. So whether you're a clinician or a patient or a policymaker or someone else, you're all welcome to join. So if you're interested in joining, you can find more details on our website and register. So that's the end of my very short presentation. If you have any questions in the future, you can visit our website or write us at info@rdconnect.eu. And that's it from my side. I'm going to now pass it for Mark on his presentation.
Okay, thank you, Dorota. Hopefully I'm sharing the right screen. So do you see my my startup screen with the little guy?
Yes, I can see.
Okay. So welcome everybody, and also thank you, Dorota, for the introduction to the to RD-Connect and the RD-Connect community, to which we owe a lot. Also, this work that I'm presenting wouldn't have been possible for the rare diseases if it wasn't for RD-Connect. So yeah, we'll hope to continue with the community. So the what I'm was asked to do and will do here is to say a few words about why FAIR for rare diseases. To be honest, it was actually not. I have loads of slides and different ways to explain this, so it wasn't actually that trivial to put a slide deck together. So I also hope that we have a nice discussion at the end of the presentation, where we can explain more. And I also see some of my colleagues in the group, so there's others who can help me with the answers as well. So what I wanted to start with is, oh, I'm not going anywhere. Yeah, what I wanted to start with is a student who did an internship with Claire Schofflin, who's one of the ERN, one of the people involved in ERNs, and she also spent a little bit of time here in our group. Very bright student, and she was doing research on HHT, hereditary. No, I'm not going to. I won't be able to express that to pronounce that, but she was doing research on that, and I'm not going to go into all of her research. It's in one of the vascular anomalies, and here you see some of the symptoms, and that it has relations with nosebleeds and genetic background. Some possible mechanisms that researchers here are looking into, and Lisa also worked on a hypothesis that you see here. As I said, I won't go deeply into the science, but I want to make some observations that we made when she was doing this research. So what Lisa did and could not do. So one of the things she did was that she had to manually extract data from medical records and medical data. I think she was lucky enough that she didn't have to take it from handwritten notes, but it was still a major task to get data into a shape that she could do some statistical analysis. So all in all, it was a big effort for getting a relatively small data set to work with. And that had a major influence, that's one of the observations we could make, a major influence on the statistics that you could do. So basically it was largely assumption driven. If this could happen, then maybe if we add this little bit of extra data, then we can just about answer that question. But it did affect the strength of her statistics. And it also meant that she couldn't do data-driven analysis. She couldn't just take all the data and see what that had what answers were in the data. The other thing that we observed is that it was, we didn't even think about comparing her results with data from other patients from other diseases that potentially have similar symptoms. This is all about iron deficiency. And it would have been nice if she could just ask a question for other diseases with the same kind of deficiencies, how treatments there would work, for instance. So these were some observations that hampered a student in doing some useful research. So in conclusion, the local data and other people's data were not readily available for analysis. And one thing that I think is for all of us to think about is that you should multiply this with all the researchers doing similar kind of investigations. And also in our group, we're a bioinformatics group. Bioinformaticians typically spend months on making the data usable. So coming back to Dorta's starting slide with the different silos and how to deal with that. I think usually in this situation, people define as a goal to share data and put data together, which seems, I guess nobody will look at this very strangely. But if you think a little bit deeper, then maybe this is not actually the end goal. Maybe this is actually more a means, the sharing data and putting data together, because the actual goal is that we should be able to analyze data and answer questions, like we would hope that Lisa would do very efficiently. And that would be for questions like this. I've just put a list of what we call driving questions for our verification cases that we do. What are differences in age loss of ambulation as a result of steroid use in DMD patients? How do patients with HCT, that relates to the disease that Lisa was investigating, how do they respond to radiotherapy in other hospitals? And what associations with iron infusions for other patients do we see? What is the current information about course and treatment of vascular malformations across several registries? What number of fractures is the most common in patients carrying an osteogenesis imperfecta mutation across Europe? What associations, more on a molecular level, for genetic variant, disease severity, and phenotypes in Rett syndrome? And where can I obtain tissue samples for transcriptome analysis, so samples to start doing some analysis on? The common denominator here is that all of these questions require multiple data sources to answer them. And then the question we could ask is, why is this not easy? Why is it that it's such a problem? And a possible bottleneck that we can think about, that maybe the biggest bottleneck for data sharing, is the actual need to share data. Because across countries and institutes, this is a problem. So if that is, so the need to share data, but maybe it's actually the perceived need to physically share data. And some of the signs of that are that people spend a lot of time on data sharing guidelines, embargoes, publication embargoes, data access forms. Debates about common data elements are a sign of this. Aligning policies between institutes and across borders, and data warehousing attempts are all signs of trying to overcome this. We do, however, have an absolute need to enable this analysis of resources, especially for rare diseases, because we do not have these data silos in one location. We have them all across the world, and because rare disease data is sparse, we would need to do analysis across them. So data is sparse, highly distributed, it's heterogeneous, it's always often in different formats, poorly interoperable, often sensitive. So there's quite a big need here. So if that sharing is such a bottleneck, then maybe we can think about a different approach. And that's what the FAIR data to a large extent is about, and also relates to the personal health train paradigm that I will talk about. So if sharing is hard, then maybe we should just don't share. Instead, maybe we should make our own data findable, accessible under well-defined conditions, interoperable, reusable for humans and computers. So that is the fair principles. So don't share, but be fair at the source. And one of the examples of where this was applied, this principle, one of the early examples, is provided by André Dekker, a colleague from us in Maastro Clinic in the Netherlands, who together with his team did machine learning or to predict whether patients should have chemotherapy or not. And he was able to show that his predictions were actually doing better than what doctors could do, and with quite drastic results. So he could show that, for instance, in Australia, where doctors assess whether people should get the chemotherapy or not, that often patients that doctors were not giving the therapy, he could predict that they often actually would have benefited from this chemotherapy. In the Netherlands, everybody gets the chemotherapy by definition, but that's equally bad because he could also predict that a lot of patients in the Netherlands that get the chemotherapy, he could predict that they would actually not benefit from the chemotherapy. And it's not a nice treatment, so the people in their last period of their life get a very harsh chemotherapy where that could have been prevented. So for this story, it's actually, I'm not going into the analysis here, but more about how he did this. Because he needed to do the machine learning, he needed data from all over the world. So that's where he applied his machine learning algorithm, but the data were never shared and were never put together. So apparently that's not necessary. And the way he did that was by making the data at each of the sources. So he had people from his team go there and make their data what we would now call fair, and especially interoperable, preparing the data for analysis in the same way, applying the same standards at each location. And that allowed him to do the machine learning algorithms on site at source and then pull the results together. And the results are not sensitive at all. It's only that the source data is sensitive. And that is the basis for the what we call the personal health train. I'll come back to that. So first, also our little demonstrator. So this is very advanced machine learning, but you can also do more of the typical questions that I showed before. So here's one example: where can I obtain biosamples of donors with an abnormality in head or neck, and which biobanks can I find these samples? And we put this into a demonstrator using the same approach that André used. So here you see this demonstrator, and I select one of the questions, get the number of biosamples from donors with a specific phenotype and from a specific region. We can type that into type in the variables. So Pistoia is the region, the abnormality of head or neck, and then process this information, and then you get the result. And here you see a table with the requested results. So that's the first thing, it worked. But you also see that if you look at the biobank and registry columns, that you see that the information comes from different biobanks and different registries. You also see that all the information here is blue. That means everything is clickable. Everything links to something else. So for instance, you see in the registry column, Ring 14 clinical database. If you click on that, you actually go to the biobank and registry finder that Dorota mentioned that in her introduction, and you find information about this database. So that also answers the questions of where can I find it. And similarly, you can do that for the other information, the diseases and phenotypes. And also note that you see different phenotypes here and different diseases. So here you can get more information about Ring 14 disease. And here you have the for phenotypes. And what perhaps some of you may also observe is that we asked for abnormalities of head or neck, and you don't see that in this phenotype column. And here we're actually making use of the underlying hierarchy of terms, because these are all more specific versions of head or neck abnormalities. And then we can also click on one and you see more information about that particular phenotype. So the underlying technology is what we often refer to as the personal health train. And there's a nice video that explains that. So bear with me. I tried this with Dorota yesterday and it should work. Oh.
Marco, can you increase the volume of the video?
Because the data are produced and managed by various healthcare providers, authorities, and more by citizens themselves. These stakeholders collect and manage their data in different ways, making the data hard to find and use. Furthermore, personal medical information is very privacy sensitive. As a consequence, personal health data cannot be used easily by citizens themselves, by physicians, and by researchers. The personal health train goes to the root of this problem by building fair data stations. Fair protocols ensure that data are findable, accessible, interoperable, and reusable. These data stations are connected by tracks, which are strictly secured and protected. Fair data trains are constantly monitored, and only trains with the appropriate validation may enter a particular station. To secure privacy, every data station has rules. Each data owner has access to his or her secured station and can easily request and control one's personal information. In addition, the owner can set rules for who can access the data and how it may be used. Communities with common interests, like patient societies, can choose to connect and organize their information through umbrella stations. With this access, a researcher can learn, for example, which cancer patients benefit most from novel therapies such as proton therapy. Or a citizen with a specific variant in his genome can be automatically alerted when new research results, insights, or possible risks become available. In this approach, the information is accessible for research, prevention, and education without the data ever leaving the station. With the personal health train, health data becomes available whilst individuals and institutions maintain control.
So I hope that that was audible. What was it?
Yes, I'm going to paste the link to this video in the chat right now. So in case you want to rewatch it later, you have the link to click on.
Yeah, thank you. So what we saw in the video, I want to focus on one thing that we saw in the animation, is the fair data station. So what is this fair data station? So it contains the data at the source, and what I think is interesting for our dealing with data sharing is that it's not limited to a common or minimal data set. It's just the data that you have at the local station. It exposes the description. It exposes the description of the data to be findable. So this description is not necessarily very privacy sensitive. It's just a description of the whole thing, and it makes it findable. it controls access. The content, the inside of the FAIR data station is prepared for analysis across FAIR data stations. So this is the interoperability part. And finally, the content is also richly described for efficient and correct reuse. So that's sort of an extension of the interoperability here. So that is the basis of a FAIR data station, and that is what we have in mind for the FAIRification in the rare disease domain, to increasingly produce more FAIR data stations, rare disease FAIR data stations. So independently FAIR rare disease data stations. We're doing some pilots. That's the first ones I'm showing here of where we're working on that. There's also some of bigger resources that have gone certain steps in towards FAIR and machine readable and interoperability. I show a few here. So EBI has a platform, that's the European Bioinformatics Institute. Orphanet has an interface that is producing interoperable data. A big protein database does so. We have a drug resource that made several steps in this direction, and there are a few more. So in the end, you will have an ecosystem of FAIR data stations, and that would allow someone like Lise to do her analysis much more efficiently and actually do good statistics on her data without spending a huge amount of time on making the data usable for her analysis. So instead of making a massive central database, which will come with massive sharing issues and several other issues that I won't go into right now, and it's usually for common data elements only, elements that are not sensitive, elements that can be shared. So we're not doing that. Instead, what we're doing is we're pushing out standards, protocols, support services to these different sources to enable this FAIR data station ecosystem. So how do we get there then for your data? And I'm not going into all details, I'm just giving you a glimpse. And one thing to realize is that FAIR is requiring that the 4 letters, that they are human, that are for humans, so FAIR for humans, but also FAIR for machines, findable, accessible, interoperable, reusable for machines. And we need that because then we can help computers help us. And that's typically quite a hard process, although doable. And one thing to think about that for people who are not so computer-minded on this call here, because I know some are, is one thing to look at it is to in analogy, to think of Mr. Spock. I don't know if all of you know the character, the Star Trek character, but Mr. Spock is born on Vulcan, and everybody there thinks very logically. It has to be logical. And in thinking of what a computer needs, you should actually think of baby Spock. So computers are like little baby Spocks. So they know nothing, they can learn a lot, but everything has to be 100% explicit, logical, and unambiguous. So that's what we will have to work on for everybody's data. And just to give you a glimpse, so this is some example data, all scrambled and such, but example data from one of the workshops that we organize. And here you see on the top, you see from 4 different sources for what we as humans easily see is mutation information. But you also see that they have different column headers. And in some cases, we're not entirely sure, but in some cases, it seems that they have a different way of describing a mutation. So for baby Spock, this would be horrible. This is not logical at all. And you also see in one case, the 4th column, that there's obviously a mistake. And as human beings, it would be easy for us to fix that mistake. But for baby Spock and for computers, it would be much harder. You should also think about scaling. So if we have to do this for 4 data sets, that's maybe doable. But if we have to do that for tens or hundreds, that becomes undoable. And at the bottom, you see data from one table. And here I want to highlight that the relationships between the values in the columns is not necessarily clear to baby Spock, because it's not specified how this W1 relates to this WFS1, how patient relates to gene A1. As human beings, we can come up with an interpretation. There's a risk, by the way, that we have the wrong interpretation. But for baby Spock, he wouldn't be so happy. It's even worse for real computers, because what we could see here, this is the human readable version. For a computer, it would look more or less like this. Some text, some different type of text, some other text. It's not very readable. So we have to make this readable for computers. And the way we do that, and that is how we set up the rare disease data linkage plan a couple of years ago with the help of RD-Connect, to do this in interdisciplinary collaboration. Because for this, you need people who know about the data, who know what everything means, who can interpret what I just shown in this previous slide. But you also need people who know about how to make data machine readable. And the people who know about machine readability, they don't know what the data, what it biologically means. And the people who know what it biologically means don't know how machine readability works at the start. What is very important for our data linkage plan is that knowledge exchange takes place. So we have our FAIR data stewards, and in every verification process, we want the domain experts, who could also be database maintainers, for instance, but that they also learn about the same process and learn about the ontologies that we use, etc. The process that we defined is depicted here in 7 steps. I won't go into the individual steps right now. There's other places to do that, but if you want, I can elaborate later. And we also put this as an Elixir deliverable, for instance. I just wanted to show you very shortly what happens when we do this. So here are 2 registries, and whoa. So PowerPoint has crashed on me. I have to start it up again. Sorry about that. Where was I? Can you still see my screen?
We can see you, but not your screen yet.
Okay, so I need to re-share my screen. Yeah, this one. Can you see it now again?
Yes.
Okay. So here you see 2 registries, and I think it's quite, so actually there's interesting information to be gained from the combination of these 2 registries. I know this because I put these slides together, but I think everybody agrees that you cannot really quickly see that already now. So integration at this level is quite impossible. This would be the situation that Lisa is confronted with. So the next step is then, is that we have to make this, and now think of baby Spock. You see on the human side that we already made the information from these 2 registries very explicit and very logical, so to say. Subject has disease, ring 14 disease, and has phenotype, salam seizures, and we also do that for the other registry. We still can't see at this level. We can't see the linking point yet. From that, we also do the machine readable part that you see with all these codes, and that's the machine readable version. So as humans, we don't have to be able to read this, but a machine can handle this. all these codes you see here, they actually come from ontologies. And what is so interesting about ontologies is that they are for humans and computers. They have these both, these two phases already. I will come back to that. I'll show an example. These.
Sorry, Marco, will you also say what an ontology is?
Aha. I'll show that in the next slide. Yeah. Then I'll explain that. Then I have an example. What is the other thing that is important is that the way this is described here is in a computer language that is globally understood. And an analogy you can draw here is that this is the English. So the way we use English to communicate between scientists, there's also a computer language that is globally understood to communicate between computers. And that's what we've used here. So this information is explicit, unambiguous, logical, and linkable, exactly as Baby Spock would like it. And now we'll go into what an ontology is in this case. So you see one of these terms, OBO, HP, etc. You can see by analogy that that is one of the phenotypes. And this HP refers to the human phenotype ontology. And here you see a snapshot of that human phenotype ontology. And what you can see, and that's important here, at the bottom, you see this hierarchy of terms. And that's typically what an ontology does. It gives this hierarchy of terms. What it also does, and that's not shown here, it also defines relations between terms. So for instance, if you, there could be, this is the phenotype ontology. Phenotypes are associated with diseases. So that will be a relation that can also be defined in an ontology, that epilepsy is associated with Huntington's disease, for instance. And what is depicted here by what I enlarged is that the same information in an ontology, this term, the same information is available in a machine readable term, this code thing, and in a human readable term, this definition. So we both can use this, human and computers. Does that make it a little bit more clear what an ontology is?
For me, I hope for everyone as well.
Yeah, okay. So what we then have is that if you look at one of these registries, we have the human readable version. On the right, we have the machine readable version. What is kind of nice is that the language that we use for this machine readable version, we can represent that as a little graph. So we often call that a little knowledge graph. So here you have you can see that there's an anonymous patient, this the two brackets, has a phenotype, epilepsy. So everything here has codes, but I've put the labels for the human readable versions there. So that makes it readable for us humans, that it has a phenotype, epilepsy, and it has a disease, ring 14 disease. And this is done at the source. So this is this will be the information that is in a FAIR data station. This is the other registry. We've done the same thing. So here you see the knowledge graph that represents the information in this other registry. Again, a person has a disease, has a phenotype, again, also epilepsy. But in this case, there's a treatment for this patient. So when we put that together, like here, we can actually see, because we used exactly the same code, and why wouldn't we? Exactly the same code for this human phenotype, they actually become linked. And that's what we can exploit by doing this step towards machine readable data. So now we can say, and it's a very simple example, of course, that perhaps for this patient who didn't have any treatment, perhaps this laboratory gene would be a treatment to consider for this patient, because we found that in another registry. Now, I've done it between two registries. Obviously, as computational scientists, we do not want any limitation to the number of registries that we can ask this question to. And I'm not going into detail how that works, but we don't need, so here in this graph, I really put it together, but we don't need to physically put this together to still do this kind of analysis over these registries. So in our rare disease data linkage plan, I wanted to show this picture because it's also a very human exercise. So critical to our data linkage plan and the verification process are the people here in the middle, the data stewards, the FAIR data stewards. And they work with local data stewards. So the disease experts or the designated people who manage the data, for instance, for DMD or for osteogenesis imperfecta, they're specific experts. I've also indicated here registry software providers because we are working on that registry software becomes FAIR data generating. That would make everything much easier for everyone. So we're working with that as well. And then on the right, you see the FAIR software engineers who are working on tooling that, for instance, the registry software providers can put into their tools. So critical are the people here in the middle, the data stewards. And you see all these little L's here. So that's the aspect of the knowledge transfer. When we collaborate, the people who have the L's here will also become a little bit of an expert on how to make FAIR data. So this is an example of how we, of the rare disease data linkage plan, how we set that up. We've also done this in our own hospital for one of the cases that we worked on. And I thought it was also nice to show it there, because this could be a paradigm of how to scale this up. If we have, again, we have FAIR data stewards in the middle. And in our case here at the LUMC, we had librarians and data managers from a data management group who were participating in a verification process. And we also had people from our ICT department who were participating in this process. So we could do knowledge transfer to these people in the hospital who hopefully in the future will also be providing further help with verification, so we can move on to other verification processes where we didn't transfer knowledge to yet. So in conclusion, FAIR is about efficient and correct computer analysis across rare disease data resources. It's about being clear and explicit about what data elements mean. And it encodes data with a globally understood computer language. And ultimately, FAIR is about making baby Spock happy. And actually, in the end, the leases of this world and us as bioinformaticians. So what support is there for FAIR? I have a few slides on that. So first I wanted to show you this. So this is a flowchart or decision tree, so to say. And you see here, so the first question to ask is, do I wish my data to be more reusable? So if the answer is no, then be our guest and silo your data. If the answer is yes, then at this point in time, where tooling is not yet perfect for FAIR, we advise to consult rare disease data linking experts. And this is one of the benefits of setting up the rare disease community, because the rare disease, the RD-Connect community, because the RD-Connect community has these, contains these experts. So I put the contact information here, but you can also contact the RD-Connect community. So after you've done that, you can ask, can we organize a verification project, and can we do that now? Because it's costly. You should know this. But if you can, then you can plan a verification project, and you can start verifying your data. And it takes roughly between 4 to 8 months. It depends a little bit of how large and complex your data set is. And it typically takes, as I mentioned, this is a very interdisciplinary process. So it takes about 3 or 4 people to be involved. Not necessarily all the time, but you need a little team. So if the answer is no, you cannot do that right now, then you can ask, maybe you want to learn more about it first. And there are a couple of training workshops I will mention one especially, the Rome Summer School, which is always very nice to do. That's 2 days. You can also, you can also recode some of your data. That's already a step towards FAIR. For instance, the OrphaNet rare disease codes, if you start using that, these human phenotype ontology codes, if you start using that, then you're already making a step towards FAIR. Or in general, structuring your data helps for a later step towards FAIR. So here's just a snapshot of such a FAIR project blueprint of this multi-month project towards verification. And this is something we, in this consultancy, that we discuss about what the costs are and what you would need and where you can get it from. There's also supporting infrastructure, and I wanted to start with what I find the most important one, and that is the community itself, the domain experts. So that can be patient representatives. It can also be the experts now combined in RD-Connect, because they have the knowledge of what the data means and what should be done with it, and that's vitally important. To me, a vitally important part of the infrastructure. Then there's the more technical infrastructures that are collaborating, and sometimes we from the rare disease community make them collaborate, which I think is a nice effect we have. The EGP, actually, this is an older slide. I didn't put the European Joint Program on it yet. That will start next year. That will be added to this then. And one very important one that I wanted to mention especially is Elixir, because that is an infrastructure that is really for the long term. And it's based on national nodes have set this up. So the countries of Europe, it's not necessarily a European Union thing. It's the countries of Europe who have joined forces to set up services across Europe. And they've also set up these platforms, what you see on the left, these 5 platforms. And you could see that as a kind of consultancy for harmonizing how we do things in Europe when it's about data. So Elixir is a life science data infrastructure. So as one of the things we do to help implement FAIR is developing tools and support. And here in this picture, I've put it in 3 layers. So the top one are really tools that we develop. Some are tools like the Data Stewardship Wizard is a tool with a web interface that you can really do as a user. Some, like the FAIRfire, would require collaboration with a FAIR data expert. But there's also tools like the FAIR Data Point that is really software that goes under the hood of another tool. So that is not for end users, but they're all software tools. There's also more human exchange. So that is doing courses. We have something called Bring Your Own Data workshops. One is associated with the Rome Summer School. We sometimes organize hackathons where we put programmers together and also do knowledge exchange there. And then finally, there's the level for the data stewards who run FAIR data projects. And we do this all for rare diseases under the umbrella of the Rare Disease Data Linkage Plan. And under in the years of the European Joint Program, we want to develop this into a service for the community that is also partly sustained by the community itself, for instance, by the ERN. And finally, we've recently are in the process of setting up a rare disease GoFriend Network. And GoFriend stands for Globally Open FAIR. And this is really a networking exercise. So what we put together is, at the moment, is a small group of volunteers who want to help foster the adoption of FAIR principles in the rare disease community, and providing hope for stakeholders, guiding the transition. So it's a small group of volunteers to help the larger community. And at the center, we have what we call handlers. That's a small group of volunteers. And they each communicate with one or more liaisons who are really working in the field. And in some cases, the liaisons come from networks themselves, like the ERN would be a network. But also data stewards, we hope, will form their own network to start helping each other to manage data more properly, including FAIR principles. So it would be interesting for us to know if you would like to be one of those liaisons or be otherwise involved in this GoFriend network. So for the future, we want to address the FAIR principles more fully. Yeah, for those who are not so technical, the metadata term is always difficult. It's data about data, but you can structure that as well, and we want to do more work on that. There's FAIR metrics, so there's people working on how to measure how fair a resource is, and that can also be an aid in becoming more fair. Especially interesting for health data is automating LC procedures, the procedures for ethical, legal, and societal impact, so consent codes, etc., consent checking. And that will also be in collaboration with infrastructures like Elixir. And filling in gaps for analytics. So FAIR will help a lot, but probably not everything, so we need to see where the gaps are. Then the other activity is for the future is scaling up. So as I mentioned, we hope to set up a FAIR data steward network that people will start helping each other. Annika, someone in our group, is working with our other colleagues on more FAIR guidelines. It's quite hard to be completely fair, especially on the machine readability side. So we would like to put out guidelines on how to make steps towards FAIR that is already very helpful. We want to publish on that. And then there's the activity to put FAIR data generation into tools that are generally used, like registry software. And this is, we already have started that in collaboration with some of these registry software providers. The approach will always be to do this through real-life cases in interdisciplinary collaboration. That's the essence of the rare disease data linkage plan, that it's always on real cases. So this is what I wanted to mention lastly, the summer school in Rome. That's always very nice. It has limited space, but keep an eye out for this, because this brings medical doctors, patient registry, patient representatives, and FAIR experts together. And the whole summer school, people learn a lot about how to set up a good registry. And that was it. Thank you very much. And I'm open to questions.
So maybe I can start. You've mentioned that making your data fair is quite time-consuming and costly. So I've been wondering, you mentioned, for example, you need to hire 3 to 4 people to work on that. Is that something you, once you do that, do you need them to keep working on FAIR forever, or is that just in the setup phase?
Oh, so that I guess that depends, and also depends on the source. So first of all, it doesn't necessarily mean that you have to hire people. It can also be that you have people working on this or doing data management in some form already, that they just can learn this extra piece from our FAIR data experts. So then it will be sort of an in-kind contribution in a way to a FAIR project. I think, to be honest, every database will require some maintenance. So the first step is the biggest step, for sure. But then when new data comes in, you will have to go through this procedure again, but then with it will be less work.
Mm-hmm. We have a question. How do you set up a new registry to be verified? Have you published guidelines?
Yeah, so partly we're in the process of doing that. So we recently put out a deliverable for Elixir, and we're going to work this up to a publication. That's a guideline. There is a website. So the GoFriend office, for instance, has a website, and Elixir also provides guidelines. I think in general, in my general advice would be to contact people. That's I think at this stage, it's still the best way forward is to discuss this with experts.
Right. So for this, you would need to contact that email, Fair Elixir. Can you remind the email?
Yeah, it was on one of the slides. Where was it? Yeah, there it is at the very bottom.
Yeah.
Or the info at
RD-Connect and EU.
Yeah.
Okay. Another question. How are the FAIR principles applied in the rest of the world? It seems more a European plan.
Oh, it's actually extremely international. So the NIH in the US, you would find it under the Commons name. They call it the NIH Commons, and that actually refers to the FAIR principles as well. What you do see is nuanced differences in the implementation, or actually it's not so much different, but it's the focus is sometimes a little bit different. So here in Europe, we spend a lot of time on linking, making data linkable. That's definitely something we put forward here. In the US, I think there's a slightly more focus on ontologies. And that's actually a nice combination that we can combine that. But for instance, one of the task forces that we set up through, partly through RD-Connect is an international task force. So there's one of the resources that's applying similar strategy is Monarch, for instance, where you have the mouse phenotype database. Applies similar principles.
And do you know if funding agencies support data verification, for example, by giving grants to different types of databases to verify their data?
So what is? I wouldn't say that well, actually they do. So there are several funders that put FAIR into their calls. I think more and more actually. I think at some point you will hardly not find it anymore. So IMI, for instance, is the Innovative Medicine Initiative has specific calls for verification of its own resources. What is more, and I'm not unhappy about that, is that what they now almost do is ask for data stewardship and data management plans. And I think it's a good thing to put FAIR principles in there. Also, if the funder doesn't ask for it, it's good to put it in there. And an very easy way to get to be more fair is to actually put it when you, for instance, have to buy a software platform, is to put it on the requirements list for that software that they need to become more fair. We've done that at the LUMC. So our data management for clinical data, the data management software that was acquired, they have now the obligation to become a more fair tool. And that's very easy because it's just you just put that in the requirements of your tender, for instance. But also reaching out to other experts to do some of the data management for you. That's we should also see here in the Netherlands, but I think internationally as well, that it's more and more required that you allocate some of your budget for data management. And then this advice would be to do that together with data management groups who are experienced in data management.
Right, and I think it's also important to mention that in case of rare diseases, the FAIR guiding principles have been have received the label of RD recognized resources, so the International Consortium for Rare Disease Research.
Yeah.
basically states in this way that fair guiding principles is the way to go and wants to draw attention of rare disease researchers that that's something they should consider.
Yeah, thank you for mentioning that, Dorota. And that actually is a reference to a guidelines document. But that's also where I make the statement to contact experts, because this is a document that data stewards and data scientists can read and understand. But I think for the typical registry manager, for instance, this is still one step too far to understand what the 15 principles there, what they mean. So you would need to have some help.
I just see we have a question from Noria. She asks, have patients to consent explicitly to make their data fair? That's a, I think, good question.
Do patients?
Yeah, I think it means like, do patients have to give consent explicitly to make their data fair?
Oh, that's actually a very nice question. We're working on that. You can imagine that for the personal health train, this paradigm of the animation, it's essential that we can check this consent. Otherwise, we'd be breaking the law. So the answer would basically be yes. And one of the things that is ongoing work at the moment is there are consent ontologies, and there's consent codes from the Global Alliance. And yeah, we need projects where we actually put this to the test, and whether we can put this into the technology of fair data stations. But in principle, a fair data station should then do that, and patients, the ideal is that patients just provide consent as they would normally do, but that we have a machine readable equivalent of that information, so that we can do automated checking. And possibly also involve the patient in the process. So for instance, they can via their phone get a message, some scientists want to use your data, is that okay? That's, we're not there yet, but this is one of the paradigms we're working on, like the personal locker, that everybody would have a locker that is basically a fair data station, but then one really personal for your own data.
But I think now we are at the stage where we need patients to help us to advocate and push that fair data become a standard, so that it's supported also on the highest levels by policymakers and funders.
Yeah, exactly. Yeah. And typically patients are our friends in this, because they see the need to make their data usable for scientists in the most controlled way, and that's what we're after here.
I don't see any more questions. So if there are no more questions, I think we can finish the session. And if you have other questions in the future, please contact us at those two addresses. So info@RD-Connect EU or fair-rd-info@elixir-europe.org. Great. Thank you very much for attending. And Marco, thanks for an excellent webinar.
Okay.
Thank you
all very much. And I'm looking forward to responses.
Yeah. Thank you. Bye.
Bye-bye.